Today we're reviewing Mendelian genetics and non-Mendelian genetics, which is a foundation for understanding how traits are passed on from parents to offspring. This review will cover Mendel's laws, Punnett squares, and more complex inheritance patterns. So let's jump into this review video that gives a quick overview of all these different components. So, Gregor Mendel was an Austrian monk, and he is often termed the father of genetics, and he used pea plants for his experiments, which helped him uncover the basic principles of heredity. So, why pea plants? They're great for experiments because they come in many varieties with traits like purple or white flowers, and plus they reproduce pretty quickly as they're R-selected species, so you can track their traits through many generations pretty quickly. Mendel focused on characters, which are heritable features like flower color or seed color, and each character has traits such as purple or white flowers, and these observable traits definitely make for an ideal experimenting organism. So here's how the experiment worked. Mendel started with true breeding plants, which were part of the pea generation, and they always produced the same trait because they were typically homozygous dominant or homozygous recessive. And he crossed these to produce a hybrid F1 generation, which would all show the dominant trait. Then, when these F1 plants were crossed, the recessive trait would reappear in the F2 generation following a 3:1 ratio, suggesting that the recessive allele hadn't necessarily disappeared within the F1 generation, but was rather being masked by dominant alleles. And so from this, Mendel developed two key laws, the law of segregation and the law of independent assortment, both of which we'll explore in detail. The law of segregation states that an organism carries two alleles for each gene, one from each parent. These alleles separate during gamete formation so that each gamete carries only one allele, and this would disprove the old blending hypothesis that was commonly believed, that hypothesized the traits would simply mix together, almost like paint. So what is an allele? It's an alternative form of a gene. For example, the gene for flower color has a purple allele, big P, and a white allele, little p. A Punnett square helps predict offspring genotypes and phenotypes. For example, crossing two heterozygous plants, big P, little p, by big P, little p, would yield a genotypic ratio of 1 big P, 2 big P, little p's, and 1 little p. However, the phenotypic ratio would be 3 purple, 1 white, as 1 big P, and the 2 big P, little p's would both result in purple flowers. So there is that whole idea of the presence of one dominant allele causing a dominant trait to be expressed. Here's some important vocabulary terms that you should know. Homozygous simply just means two identical alleles, which would be like big P, or little p. Heterozygous means two different alleles, so big P, little p. Genotype is simply the genetic makeup, so that would be like one of those big P, little p, all those things, whereas phenotype is the observable trait that results from the genotype. So whereas big P would be the genotype, purple would be what we consider the phenotype. And you need to make sure that you understand these, as these terms definitely appear quite frequently within genetics. If you don't know whether a plant showing the dominant trait is homozygous or heterozygous, a test cross can be used by crossing it with a homozygous recessive plant, and we can see how the offspring show the recessive trait and how to determine whether the unknown parent is heterozygous or homozygous dominant. If there are offspring who are showcasing recessive traits, we can infer from that the dominant unknown genotype must be big P, little p, just because it would have to be passing down little p for it to have a recessive offspring. So, the law of segregation applies when tracking a single trait known as monohybrid crossing, whereas the law of independent assortment applies when tracking two traits simultaneously through a dihybrid cross. And the law of independent assortment says genes for different traits assort independently of each other during gamete formation, assuming that the genes are different chromosomes. This means that the inheritance of one trait, like seed shape, doesn't affect the inheritance of another trait, like seed color. In a dihybrid cross, the phenotypic ratio of offspring is always going to be 9 with both dominant traits, 3 having dominance for trait 1 and recessiveness for trait 2, 3 with recessiveness for trait 1 and dominance for trait 2, and 1 with both recessive traits. And this is assuming that Mendel's law of independent assortment is in action, that a dihybrid cross between two heterozygous parents would result in this ratio. So, In terms of probability, which is a common theme shared within the law of independent assortment and law of segregation, we have multiple different rules. One rule is the multiplication rule, which states the probability of two independent events occurring together, which would equal the product of their individual probabilities. So if you see the word both, that's a key sign to use multiplication rule. Whereas the addition rule would state that the probability of one event or another mutually exclusive event occurring is the sum of their probabilities. So just seeing the word either or it should be a giveaway to use the addition rule. Mendel's laws explain many patterns within inheritance, but some traits follow non-Mendelian inheritance, which is what we're going to be covering next. Incomplete dominance happens when neither allele is completely dominant. The heterozygous phenotype is a blend of the two homozygous phenotypes. So, for example, red and white flowers would produce pink flowers, which again ties back to the blending hypothesis that Mendel's experiment rejected, just because he was using pea plants, which happen to follow a dominant recessive pattern, whereas there are definitely various different amounts of inheritance patterns among species in the world. Codominance occurs when both alleles are fully expressed in the heterozygous individual. A classic example of this would be blood type, such as AB, which shows both A and B antigens equally. Some genes have multiple alleles within a population. For example, coming back to blood type, we have the A, B, and O blood group system, which has 3 alleles. Pleiotropy is when a single gene affects multiple phenotypic traits. An example of this would be sickle cell disease, which affects red blood cells and other body systems. On the contrary, we have polygenic inheritance, which occurs when multiple genes influence a singular trait, which would lead to continuous variation. This could include traits like skin color and height, which, again, we can see within our population there's a lot of variance, considering there's multiple genes that influence this. Genetic linkage just means genes that are located close together on the same chromosome would tend to be inherited together, which would be an exception to the law of independent assortment that Mendel proposed earlier. So obviously, if the genes are closer together, that would mean crossing over would not separate these linked genes as much. And that completes our review of Mendelian and non-Mendelian genetics. Remember, understanding these concepts will definitely be very important for AP biology. Good luck and thanks for watching.